Hello, welcome to Eurofins exhibitor session for 2021 SOT. My name is Brian Kosey. I'm the Scientific Director of Ion Channel Services, and I will also be co-presenting today with my colleague, Ellen Berg, Chief Scientific Officer. The topic that we would like to discuss today is HTS and cell-based screening approaches for identifying potential cardiac liabilities. The aim of this session is to give you some information about HTS screening, but also using cell-based assays to detect potential cardiac safety issues and also cardiotoxicity. The topics that we will discuss today and the first half of part of my presentation will be the evolution of ion channel screening, also looking at EFA's platforms, and specifically I'm going to focus on the Cube 384. And I'm going to look at that approach from both safety and both HTS assessments. And my colleague will be covering cell-based assays, looking at Phenotypic and Biomap services that we provide, and some of the background for those as well. Before I address HTS testing, I thought it would be important to look at the evolution of ion channel testing through the last 20 years. As many of you may well know, manual patch clamp has been the gold standard for assessing ion channel pharmacology. It provides a high fidelity recording. However, the throughput for manual patch is very low. So to drive drug discovery projects or look at safety de-risking, it's not ideal for assessing a large amount of compounds. Thus, there was really a need to be able to test more compounds early on in drug discovery. So there was lots of evolutions that came along in many platforms to try to address this issue. And the one that I'm going to focus on is Ironworks, which this now brought from 1 cell per experiment to a 384 well platform. So this allowed now to test 550 to 100 IC50s per day versus possibly only 1 IC50 if you were running it using manual patch. However, there was some sacrifice for this increase from 1 to 384 in terms of the fidelity of the recording. Since Ironworks was a population patch and a perforated patch system, it didn't always correlate really well to what we would see in manual patch clamp in terms of IC50s. However, as evolution progressed, we moved to the QPatch. This is now, we have a 48 well platform, but we've recovered a lot of the fidelity that we had in the manual patch. So there's a great correlation to QPatch data to manual patch in terms of looking at IC50s. And we've also increased our ability to look at 25 to 50 IC50s a day versus the 1 using just manual patch. So basically, there's been a huge change in the ability to be able to prosecute many more compounds earlier on in drug discovery to be able to assess safety issues. So this leads us to how does the first gen QPatch go to the Cube 384? And this was a monumental change. We now had a huge increase in data points and also the ability to drive pharmacology earlier on in drug discovery, but now be able to assess in a 380 well format versus a 48 well format. The other big advantage of going to the 384 platform is the cost and also the time that it would take to run these assays as well. Now I'd like to provide you with an overview of the Cube 384 platform and really show you the differences, one between the QPatch, which was a 48 well platform, which now the Cube is a 384 well platform with minimal loss in fidelity. So different from what we saw from Ironworks to QPatch, where there was a monumental increase in fidelity, we are now going from a 48 back to a 384 well format that can provide us the same fidelity that we had in the 48 well QPatch mode. So obviously going from 384 to 48 is a monumental increase in data points per run. As I mentioned in my previous slide, there's a much lower cost for consumables because the 384 plates cost about the same as the 48, so you're now getting an 8-fold increase in data points for the same amount of consumable cost. The other advantage to using the Cube is now we have temperature control, so now you can run at physiological temperatures or maintain a constant temperature throughout your recording. It can be true walk-away operation. Now we can run up to 6,000 data points without any intervention by a scientist. And it also allows for now true HTS screening. Now that we can run more than 1,000 compounds per day easily in single point or dose response. The other advent that has increased the ability to use the Cube as a HTS platform is it has a very powerful online analysis and assay metric filtering. So it's almost impossible to review every trace from a 384 well run, but now you can use metrics to be able to screen out the data that you don't want and only export data that meets your criteria. The current slide I'm showing you is basically an overview of the ion channel targets that we have transferred to Cube at Eurofins. So these are all the recommended SIPA targets from HERG all the way to KIR 2.1. And these are the major ion channels responsible for the depolarization and repolarization of the action potential in the heart. And basically we've created two offerings from this. One, our cardiac profiler core. So here we're looking at HERG, NAV, and CAV. But also the full panel as well that includes the addition of KB 4.3, KCNQ, KIR 2.1, and also NAV 1.5 late current. And this is being offered as both a single point assay and also a dose response 6 concentration assay. So how do you use these assays in order to support your drug discovery projects? So this is just a generic schematic of how you could approach using different assays that are on the Cube in order to de-risk cardiovascular safety. The advent now of a 3D4 platform, you can now do true tier 1 safety screening, both looking at HERG and possibly even a cardiac profiler of looking at all 3 channels early on. And the reason this is advantageous is, one, you can have early hazard identification of your compounds, but also this can be done in a cost-effective, efficient manner that was not possible before. But as your project progresses into lead optimization, now you can start looking at more dose response effects or also dose response effects of multiple channels. And again, this can build optimized data for your SAR models and really drive your chemical design to provide a compound that does not have safety liabilities. And then in the final stage, when you're at candidate selection, now you would want to address looking at full panels so you could get an overall holistic idea of what your potential safety concerns could be once you move to in vivo models. And also you can look at an integrative approach as well, looking like at an MEA or an IPS type of assay to correlate to what you see in that cardiac full panel. So the offerings that we have at Eurofins, and we just launched our HERG assay as of January 2021. As I mentioned previously, we have a core and full panel, which both of those were launched early this February. And we also have the ability to look at functional target-based ion channel discovery. And we also have that available as of January 2021. And in both our HERG and both of our panels, we can again run those in single concentration or dose response with 6 concentrations. This slide is just referencing some of the reference pharmacology that we generated on the Cube 384 platform. And I don't necessarily want to highlight the IC50s, but I wanted to show for 3 individual targets, HERG, KB 4.3, and CAV 1.2, that there's great reproducibility from run to run. And if you can look at HERG there for E4031, there's 3 independent traces as well as with verapamil, and the overlay of the dose response curve is very consistent. The same goes for CAV 1.2 for nifedipine and also for flecainide and quinidine as well. And again, as I talked earlier in the talk about the loss of fidelity, in this case, again, moving from 48 well platform to 384, the fidelity of the recordings and the reference IC50s are kept intact. This slide just illustrates the reproducibility that I showed you on the previous slide by looking at multiple runs of verapamil. So over 11 runs of verapamil, the IC50 is less than twofold in each instance, and the Z primes are all greater than 5. So again, this shows the reproducibility of the assay over multiple runs, and that you have confidence that you're going to generate high fidelity IC50 recordings with this 384 platform. Moving on from safety screening, I thought I would provide you with several examples of HTS screening to support drug discovery screening efforts. And in this particular example, we can look at both a parental line, we can also look at multiple mutations, and also the native channel. And by using channel biophysics, we can potentially develop a protocol for a screening effort. And this allows us to characterize the cell lines early on in a high throughput format, so we can provide all that data early on in a quick and efficient manner. Once that protocol is generated, now we have the ability to develop a screen to look at tens or even thousands of compounds on multiple cell lines. And in some cases, if there are multiple mutations or multiple cell lines we want to address and they use the same voltage protocol, we can assess all those on the same plate. On the second example, this is looking at multiple subtypes of one particular channel. So if we look at, again, for an example of a channel that's 1.1, 1.2, 1.3, we can look at all those channels simultaneously now that we have 384 wells that we can distribute these different cell lines through. So this allows for an early on ability for selectivity of your compound. It can also be done very efficiently. And one of the biggest things when you're doing early on screens is compound is at a limited resource, so you could provide this all on one plate. So once you have an idea of what your compound selectivity is, you could do further dose response screening, and also, as I mentioned earlier, some of the safety assays as well. And again, this can be done on 10 to thousands of compounds very easily in a both cost-effective and efficient manner. Besides the safety targets that we offer, we also have over 60 plus ion channels in our portfolio at Eurofins. These come in both ligand gated and voltage gated, and these two cells are just to provide you an idea of the diversity of ion channels that we can offer. And we can offer these both as screening, but also as potential targets as well. And any of the ones that are not indicated on these slides, we also have the ability to generate custom cell lines as well. So we can support drug discovery needs from both the initial screening for certain targets, but also as looking at counter screening and also further on profiling of your chemical series in any point in the drug discovery process. So I thought I'd just highlight again what the main points are for the Cube utility. Obviously, the first part of my talk focused on the safety, and really what the 384 platform brings us now is efficient and throughput running safety panels. So turnaround time of assays is greatly enhanced, and it can also be done in a larger format. The other thing is we have the flexibility to run single point assays as well. Since you have 384 well to work with, that the ability to run single point assays is much easier than in the legacy QPatch, where you only had 48 wells. And it can really support the expanded testing in Tier 1 screening. And that's not only HERG, but other cardiovascular safety ion channels as well. And this can be done in a cost-effective manner. But the other part of the second part of my talk was looking at target. So again, looking at HTS capabilities for drug discovery projects. We now have the ability to support Tier 1 or actually primary screening of targets in terms of ion channels, or also selectivity assays as well. We can do primary functional screening. And again, the flexibility of the Cube platform, both in assay design and screening design, is very flexible in terms of supporting any types of needs that you would need, any types of needs for your projects. And this is both, again, we can do it at single point and also DR follow-ups for any of your screening campaigns. To sum up the portion of my talk, I thought I would just highlight some of the benefits of running the Cube 384 platform at Eurofins. Just as a reminder, Eurofins is an industry leader in high-quality ion channel cell lines and services. We have technical expertise to support all of your project needs. We have over 60 years of electrophysiology experience and have helped thousands of clients in supporting their drug discovery needs, both from a safety perspective and also primary screening as well. We have a broad range of platforms. We're continuously making investments in innovation and having the latest and best platforms to be able to support our clients' needs. We have a leading portfolio of validated ion channel cell lines, and we can offer rapid turnaround times or expedited turnaround times. And also, we are cost effective in being able to support ion channel screening projects. And we can do this in a multitude of different ways, both from single point all the way to dose response, and in both cases allow for rapid, low cost, and large volume screening to support your drug discovery needs. This ends the portion of my presentation. I would now like to hand it off to my colleague, Ellen Berg.
Thank you, Brian. Now we're going to turn our attention towards mechanisms beyond cardiac ion channels that contribute to cardiovascular-related toxicities, mechanisms that we are addressing through phenotypic approaches. These mechanisms involve the vasculature, specifically thrombosis and vascular toxicity related to atherosclerosis. And we are addressing these mechanisms through our Biomap Phenotypic platform. The Biomap platform consists of standardized assays using human primary cell types that measure protein endpoints, a large reference database, and various analytical tools that support knowledge discovery of mechanisms of action and toxicity hazards. Biomap systems are built to capture a diverse range of tissue and disease biology. To maximize physiological relevance, Biomap systems incorporate human primary cell types, co-cultures, combinations of stimuli to more accurately model tissue and disease states, and they use clinical protein biomarkers to make it easier to translate results to clinical outcomes. The Biomap Diversity Plus panel is a standardized panel of 12 assays that have been running continuously for more than 10 years. They capture a broad range of tissue biology of the vasculature, the immune system, fibrosis-related biology, and respiratory biology. Assay conditions reflect adaptive responses, the biology that's active in all of us. Here is an example of a drug profile tested across this panel with peaks and troughs representing different biomarker changes. So what can we do with this platform? Support clinical translation by connecting biomarker activities to clinical effects. This is the profile of trametinib, a MEK inhibitor used to treat melanoma. There are biomarker activities that can be associated with clinical efficacy. These include inhibition of cell cycle, angiogenesis, and tissue remodeling, as well as activities associated with known side effects, increased risk of infection, cardiovascular side effects, and skin rash. We can also compare drugs to one another. Here we see a comparison of the profiles for the PDE inhibitor apremilast and the TNF-alpha antagonist Enbrel, both drugs approved for treatment of rheumatoid arthritis patients. Common activities are more likely to be important for clinical efficacy. One of the more valuable applications of phenotypic profiling is the ability to inform on mechanism of action. Searching the reference database to find the most similar profiles generates a mechanistic hypothesis. And these data are actually sufficiently robust and reproducible to permit the development of mechanism classifiers by machine learning. This is a cluster analysis of profiles from compounds with known but different mechanisms, indicated by the color, and tested at multiple concentrations, which are indicated by the bubble size. You can see how well mechanisms can be separated. Tight clustering across multiple concentrations is a feature of high target selectivity and preferentially found in marketed drugs. We have also used the platform to uncover mechanisms of drug-induced toxicities. We start with data from drugs that cause the same side effect. Then, if we identify shared activities or a signature, we leverage the large reference database as well as literature data to confirm the association. By exploiting target information on the reference compounds, we can then infer target mechanisms that are associated with each toxicity signature. Using this process, we've developed 9 of these mechanism-based toxicity signatures. For the rest of the presentation, I'm going to focus on 2 toxicity signatures that are relevant to cardiovascular toxicity. The first one is a signature for thrombosis-related side effects. These include stroke, deep vein thrombosis, and pulmonary embolism. The key biomarker activity is increased cell surface levels of tissue factor in a vascular inflammation model using human primary endothelial cells. Of course, the fact that tissue factor is the primary cellular initiator of coagulation gives it a pretty strong biological rationale. But it was data mining of the reference database that truly fleshed out the mechanism of toxicity at the level of tissue context. It turns out that the target pathways inferred from this analysis all converge on the process of autophagic vacuolization in endothelial cells. By promoting increased vacuolization, larger amounts of tissue factor are sent to the cell surface. This is a normal process that would recruit nutrient-rich platelets to a tissue site, promoting increased angiogenesis and wound healing. But of course, as we know, normal processes can be hijacked and lead to pathologic consequences, in this case, increased thrombotic potential within the vasculature. Given the recent association of thrombotic complications with severe COVID-19 disease, we became interested in the therapeutic potential of targeting this mechanism. And so we wanted to look for inhibitors of tissue factor. So we did the reverse study, looking for inhibitors of tissue factor, and we did find a few interesting target and pathway associations. For example, inflammatory pathway inhibitors, IL-1 and TNF-alpha, which were not surprising. We also identified a number of kinase inhibitors with strongest support for p38 MAP kinase and MEK as associated mechanisms, a few epigenetic mechanisms, including BET, HDAC, and proteasome inhibitors, and interestingly, we also identified insulin and calciferol or vitamin D. For each of these pathway mechanisms, here are listed the references from the literature that support possible roles of these in reducing thrombosis. Given the association of thrombosis with COVID-19 morbidity, these may be very interesting mechanisms to pursue. Putting this all together shows how mechanisms associated with increased risk and decreased risk converge to regulate tissue factor and potentially to impact thrombotic events in patients. You can appreciate from this the usefulness of phenotypic profiling for highlighting the diverse contributions of environmental factors, comorbidities, and drug history. The second toxicity signature we'll discuss is that for vascular toxicity. This signature is associated with increased hypertension and cardiovascular events due to atherosclerosis. The key biomarker activity is increased cell surface levels of serum amyloid A in a vascular inflammation model using human primary coronary artery smooth muscle cells. Serum amyloid A itself has been shown to be an independent risk factor for cardiovascular mortality, again, giving it supportive biological rationale. And following the same process I described for the thrombosis signature, we've been able to identify mechanisms that increase serum amyloid A in this coronary artery cell-based system. And as you can see here, they are all associated with increased risk of cardiovascular adverse effects. And mechanisms that decrease serum amyloid A in this system are associated with decreased risk of cardiovascular related side effects. Again, we see how mechanisms associated with increased and decreased risk converge to regulate serum amyloid A and potentially to impact risk of cardiovascular events in patients. And we see again how contributions of environmental factors, comorbidities, and drug exposure can be integrated. Now, in order for us to get the most value out of these assays and platforms, we will need a way to incorporate these results into program decision making. For this, we support the use of decision frameworks, such as adverse outcome pathways used in chemical safety risk assessment or discovery program outcome pathways used for drug discovery. These frameworks connect our knowledge about a drug and how it works at the different levels of biological organization to support clinical translatability. And you can see how we can map in vitro and phenotypic assays to key events along the pathway. Now, from our presentation, I hope you are beginning to see how human cell-based phenotypic assays can benefit many stages of the drug discovery process, providing a useful bridge between target-based information and clinical outcomes. In summary, there are diverse mechanisms that underlie cardiac and cardiovascular toxicities. Target-based and phenotypic solutions work together to cover these. Phenotypic platforms can reduce the use of animals, support clinical translation, identify mechanisms of action, and help in the discovery of novel toxicity mechanisms. And of course, decision frameworks can facilitate the adoption of non-animal in vitro and phenotypic assays for drug discovery decision making. We'd like to close by acknowledging and thanking our respective teams. We'd like to remind you of our expertise across a number of major target classes. And we'd also like to highlight our Discovery One integrated drug discovery offering, which brings together our capabilities in chemistry, in vitro pharmacology, safety pharmacology, ADME-Tox, translational phenotypic assays, in vivo solutions, and custom assays and products. For more information, you can contact us here, and we thank you for your attention.